In this video, I'll be continuing on with the cell structure topic. I'll be going through cell division and particularly mitosis. The cell cycle is made up of three phases: interphase, mitosis, and cytokinesis. During the interphase, the cell is preparing to divide. You have the G1 phase, which is where the cells grow, new organelles are made, and proteins are made. Then is the S phase, which is where DNA replication occurs and then it's checked for any errors. Then is the G2 phase, which is where the cell keeps growing and prepares itself for mitosis. Then is mitosis. So the definition of what mitosis is that it is a nuclear division which produces genetically identical daughter nuclei. A way to try and remember the stages of interphase for your exam is that the S phase is DNA replication, so that's just something that you have to remember, and the rest are Gs, which are for growth. The first G phase is for doubling organelles and making proteins, and the second one is right before mitosis, so this is when the cell is preparing for nuclear division. We have 46 chromosomes or 23 pairs of chromosomes in our cells. During S phase with DNA replication, this will then double to 92 chromosomes, and after this, in mitosis, it will split into two nuclei, each with 46 chromosomes. Now, in your exam, not only do you need to remember how many chromosomes humans have, they may ask you to apply mitosis to other animals, in which you need to double the number of chromosomes they tell you about and split those chromosomes accordingly. So, mitosis is needed for growth of multicellular organisms, repairing damaged tissues, and asexual reproduction. During mitosis, the DNA condenses and coils to form chromosomes. As you can see here, this is what a chromosome looks like. The dot in the middle is a centromere, and half of a chromosome is called a chromatid, and the other half is the corresponding sister chromatid. There are 4 stages to mitosis. The first stage is prophase, which is where the DNA condenses into chromosomes. The nuclear envelope breaks down, the centrioles move to opposite poles and start to produce spindle fibers. In metaphase, the spindle fibers attach to the centromere of the chromosome, and the chromosomes line up along the equator. In anaphase, the spindle fibers contract and pull the sister chromatids to opposite poles. Then, telophase, the DNA decondenses, the nuclear envelope reforms, and spindle fibers break down. A way to remember this in your exam is that in prophase there are 3 things to mention, and those 3 things are similar in telophase, just the opposite. For example, in prophase, the DNA condenses into chromosomes, but in telophase, it decondenses. The 3 things you need to remember are about DNA, nuclear envelope, and spindle fibers. Then metaphase, remember the M stands for the middle, which is where the chromosomes line up. After mitosis is cytokinesis, which means the division of cytoplasm. So this will separate the 2 cells from each other. The end result of mitosis is the formation of 2 genetically identical diploid daughter cells. Diploid just means a full set of chromosomes. Other organisms produce genetically identical cells, but the way they divide is different. Bacterial cells divide using binary fission, and fungus divide by budding. Finally, this entire video has been about controlled cell division, where there are checkpoints at each stage to ensure the cells are dividing as they should. However, cancer tumours are formed due to uncontrolled cell division. The reason why division becomes uncontrolled is because there are mutations in the genes that control the division, called oncogenes. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.